Bright Tuty. Every student matters.
Let's move on to the next subkingdom, that is phanerogram. Phanerogram is a subkingdom in which the plants they develop seeds. Okay, so the main difference between cryptogamy and phanerograms, they develop seeds, they have well differentiated reproductive parts. All right, the seeds consist of embryo along with the stored food. The embryo has stored food. This stored food actually initially helps in germination of the seeds. When the seeds germinates and give rise to new plants, so in the initial stages, the stored food in the embryo provides nutrients to the seeds. And later on, after it develops the roots, it starts absorbing nutrients from the soil. They have well-formed stem, leaves and roots, and they are further classified as gymnosperms and angiosperms. So next is the gymnosperm. The gymno word means naked and sperma means seeds. So these plants, they possess naked seeds. They are usually perennial, evergreen and woody. Examples are pine, deodar, cycas, mostly these trees you can see in the hilly areas. So naked seeds means seeds are not enclosed inside the fruit. Okay, they are in the cones, they are exposed. So these are gymnosperms. Next is the angiosperms. Angio means covered and sperma means seeds. So the seeds are covered. So you can very well differentiate between angiosperms and gymnosperms. The main difference, in gymnosperms seeds are naked, in angiosperms the seeds are covered inside the fruit. They bear the flowers, fruits and seeds. They are also called flowering plants, while the gymnosperms were non-flowering plants. They possess seeds enclosed inside the fruit. The seeds germinate and develop into new plants. The embryo and the seeds have structures or the seed leaves called the cotyledons. These cotyledons are called the seed leaves and they represent a bit of pre-designed plants in the seeds. Right? Angiosperms are further divided as monocots or dicots based on the number of cotyledons they have. So if you are asked to differentiate between monocots and dicots, this is how you can do. Monocots have parallel venation. The leaves, okay, I'll draw here. If you draw a leaf, so they have parallel venation, kind of. Right? But in dicots, they have reticulate venation like this. This is reticulate venation. So monocots, first difference is in the number of cotyledons. Monocots, the seeds have one cotyledon. Dicots, the seeds have two cotyledons. Second difference is the venation. The leaves have parallel venation, while in dicot they have reticulate venation. The third difference is between the root system. So in the monocot have fibrous root system, dicot have tap root system. So if you know the fibrous root system is like this. Many tiny roots arising from the base of the stem. But in a tap root system, there is one main root and some tiny roots here and there, fibers. So monocots have fibrous root system, dicots have this tap root system. So these are the three main differences between monocot and dicot. So example of monocot is, you can see, you must have seen many examples. Rice is one of the example in which the seed has only one cotyledon. And for the dicot, you have seen the beans or the kidney beans, all the pulses which have two cotyledons, they fall under the category of dicots.